I met with him a couple of times.
Okay.
And then I started in January and he left, I think, in November.
Okay.
Yeah, so I got a technician from him, which is great.
All right. We get started. Morning, everyone. Welcome to another Terasaki seminar series. Today we have Professor Sharon Gerecht from Department of Biomedical Engineering at Duke University. She's a global expert in vascular and stem cell biology and engineering, including pioneering engineering principles to understand and harness cell-fate decisions for therapeutic developments. She's received many awards. For instance, she's a recipient of Ellen C. Davis Medal from the Maryland Academy of Sciences, the North America Vascular Biology Organization Junior Investigator Award, National Scientist Development Award, Established Investigator Award, both from the American Heart Association and the National Science Foundation Career Award. She is also elected Fellow of the American Institute for Medical and Biological Engineering, elected member of the National Academy of Medicine, and elected Fellow of the American Association for the Advancement of Sciences, and the National Academy of Inventors. She has authored more than 150 papers, book chapters, and patents in her field. The floor is yours.
Thank you so much for the introduction, Mehmet, and the invitation to speak with you today. And today I'd like to talk to you about a microenvironmental regulation of blood vessel differentiation and assembly. Our lab is interested in understanding and controlling cell fate and morphogenesis. What we're trying to understand is how we can control pluripotent stem cell differentiation or fate into specialized cells. In our case, we are focusing on the vasculature. How we can control their morphogenesis into functioning blood vessels. And of course, to induce homeostasis that the vasculature then will stabilize. In our body, while we have the homeostasis, there could be extracellular stresses that disrupt this homeostasis. So we're trying to understand how we can reactivate this morphogenesis as well as differentiation pathways to try and normalize the vasculature. So when do we have these extracellular stresses? Typically during disease development, and we specifically work in cancer and atherosclerosis, of course, during injury, and in extreme conditions, which is a newer area for our studies, where we look into space radiation and different types of threats. So we're trying to develop therapeutic, but also develop countermeasures to try and prevent this damage in the homeostasis, and again, to restore function, repair, and homeostasis. So what kind of stresses do we study? We study low levels of oxygen, hypoxia. We study changes in the surrounding extracellular matrices, changes in flow and in strain, and as I mentioned, also recently radiation. So in this talk, I'd like to focus on the low level of oxygen, hypoxia, and changes in the surrounding extracellular matrices. So when we look at the microenvironment of tissue or organ, in our case, the vasculature, we're noticing different players here. First of all, the multicellular with multi types of cells with direct and indirect communication. We have a scaffolding that are either as two-dimensional surfaces that the cells on top of them, or as scaffolding. We have three-dimensional scaffolding. Of course, we have the extracellular matrix with different components of the extracellular matrix listed here, and different physical cues such as topography, stiffness, vasculoelasticity, different degradation sites, etc. There are also various physical chemical cues such as oxygen, different growth factors and cytokines, binding motifs, and of course mechanical cues, especially in the vasculature, where we have the blood flow that induces shear, and also strains on the vessels themselves. So we do a lot of our work by designing biomaterials that try to mimic this microenvironment, again, as two-dimensional surfaces or 3D structure, where we can control the mechanical strength. Those hydrogels are biocompatible and biodegradable, And the nice thing about them, they are design defined properties. So we basically can really define the structure and tailor properties so to induce function. So this is how this hydrogel turns from solution to gel. We can embed cells in them and study what happens. And we can also transplant those hydrogel for therapeutics. And I'll try to show you most of these things that we have done with those. So how the microenvironment regulates vascular assembly was the first question we asked, where we looked at both adhesion and degradation sites. And these are a pretty old study where we established differentiation of pluripotent stem cells to what we call early vascular cells that can make both endothelial cells and pericytes with these specific markers. And this is how these endothelial cells look like, and this is how the pericytes look like. And then we ask, what if we'll take these early vascular cells that compose both of these cell types that can make the vasculature, and we encapsulate them in engineered hydrogels. And specifically, we chose to use a acrylated hyaluronic acid, which has both the adhesive motif, the RGD, and also can be degraded, or it's cross-linked with degradable, MMP degradable sequence. And indeed, we make those beautiful networks. These are from the human endothelial progenitor cells, and these are from IPS derivatives. And you can see this is, you can see that, but it's 600 micron, and we can see these beautiful networks there. So that was basically the first time that human engineered vessels were formed in completely synthetic material. These networks can also integrate in the mouth once we transplant this, in this case, subcutaneously. And as we use both endothelial cells and pericytes, we could see these endothelial cells here in red acting as pericytes or as the vessels themselves. So with that in mind, we continue and look into how a microenvironment regulates vascular fate, integration, and assembly, and specifically with hypoxia, which is again low oxygen tension. Hypoxia is a major part of many of our studies, and the reason is that the hypoxic environment is really a presence during development, injury, and cancer, which are all areas where blood vessels can grow and respond to those cues. How hypoxia happening, it's when oxygen consumption by the cells or by the tissue is happening, but there is limited oxygen diffusion. Hypoxia activates signaling of new blood vessel formation, and the molecular mechanism typically involve the hypoxia inducible factors, 1 and 2 typically, and reactive oxygen species. When we talk about hypoxia, it is between 0.5 to 5% oxygen. And why we even started to work in this direction. First of all, when we started it about a decade ago, there were limited studies that looked into hypoxia and vascular differentiation. And most of the studies that were done with cancer cells and some in the vasculature were mainly using genetic modifications and gas chamber or specialized hood, which we still use. And typically they were done in 2 dimensions, either Petri dishes or more in the recent years, microfluidics. And what we did notice is real-time analysis are lacking. So we didn't understand rapid responses, and also when gradients of oxygen tension were formed, how the cells responded. So I'll walk you through kind of the evolution of the study and see how we moved from 2-dimensional to 3-dimensional and from in vitro to in vivo. So first we wanted to understand how hypoxia regulates differentiation. The cardiovascular system is the first to develop in our body. So it's developed, of course, with a passive or oxygen diffusion, not active transport of oxygen. So we said, are these conditions impacting the differentiation? So this is the differentiation schematic that we used back then. We now use a little bit more modification, but it's about the same, where we take our iPS cells, we allow them to, or we induce them to differentiate into kind of mesodermal cells, then we reset them. So this is happening in about 6 days, then we reseed them. And this here we use collagen 4. Now we know we can use also laminin and fibronectin and allow them to generate those early vascular cells. So what we did, we just did this in a hypoxic and non-hypoxic condition. In this incident, we used 21%, of course, the atmospheric condition, and you can see when we measure the dissolved oxygen just with the sensor, so it's not invasive measurement. You can see this is the level of oxygen. When we change the media, this is the level maintained of course, non-hypoxic. When we differentiate the cells in 5% oxygen, you notice quickly that the cells consume oxygen very quickly, and whoever works with IPS cells know that they proliferate really fast. So they consume oxygen. This is where we change the media, and again, they start to consume the oxygen. And the levels here that are, after a little bit less than 72 hours are lower than 2%. It's already activating signaling pathways related to hypoxia. So we said there's this impacting at all our differentiation. And what we noticed that in the 5% oxygen, we started to see these small colonies that turn to be positive for endothelial cells. When we do a flow cytometry for weak adherent, we see upregulation when we culture the cells in 5% oxygen. And this is how the colonies look like when we culture in a hypoxic condition. We could see those weak adherent positive cells surrounded basically by pericyte cells. So this is starting to look like the organization that the vasculature will have as is maturing. We then ask, is it the early stages of differentiation or later stages of differentiation that's impacting the fate? And when we culture the cells, what we call secondary, so first the mesodermal induction in atmospheric condition, then hypoxia, or the other way around, what we have found is actually very striking. We have found that when we culture the cells or we differentiate them with beginning with 21% oxygen, we don't see upregulation at all of the vascular markers. This is weak adherent and CD31, and this would be the control or the secondary. But when we culture the cells early on, either just for the first 6 days or for the entire differentiation in a hypoxic condition, we see upregulation of both of these markers. And this basically leads to a complete differentiation and maturation of the cells in either if the cells are primed with hypoxia or continuously in hypoxia. So what we noticed since that publication that was already several years ago was that other stresses also impacting the vascular fate. So hypoxia turned to be just one of them. Quinton Smith, that was in the lab, he studied how the cells respond to different stiffnesses of surfaces. And we have shown that the softer matrices actually induce vascular fate. He also actually showed that when we micropattern extracellular matrices in different shapes and sizes, we can control the fate either to differentiate only to pericytes, to their mix of endothelial and pericytes, and control their organization. So continuing back with the hypoxic study, Bria Macklin that graduated in the lab less than a year ago wanted to ask, does hypoxia regulate vascular integration? And the reason for this question is because if we'll think about therapeutic, we're going to use these cells where they are exposed to hypoxia in the injured or diseased tissue. So we wanted to see how our cells respond to this in terms of integration with the host vasculature. So Bria started with a simple experiment. She just put this, and we also use, hypoxic retina as our experimental model, and I'll explain in a couple of slides why. So she just put those cells, our IPS derivatives, and mature human retina endothelial cells as control in hypoxia. And as you can see, between atmospheric and hypoxic, we could not see really any difference. The only difference we noticed was that in our IPS derivatives, we could see that the proliferation maintained the same when we put them in hypoxia, but it's slowing when we put the mature human retina endothelial cells. We did find striking differences when we make a 3D network, specifically here in collagen gel in a low hypoxia, 1% oxygen. When we encapsulate IPS derivative, we see these beautiful networks that we couldn't really find when we encapsulate the human retina endothelial cells. And we also quantify that. Then we wanted to test in vivo. And that's the reason why we use the retina. So we decided to use to see if the IPS derivative integrates with the host vasculature in the oxygen-induced retinopathy. This was done in collaboration with Elia Dow at the Wilmar Institute at Hopkins, where I've been at the time. So what's happening, the OIR model is actually a recapitulating retinopathy. During retinopathy, what's happening is that the healthy vasculature is exposed to a continuous insult. In many cases, it's in diabetic patients. So we have high sugar, and this leads to capillary dropout. This capillary dropout creates this hypoxic condition right here, which induces overgrowth of the vasculature, or what we call the pathological neovascularization. If this goes untreated, the patient can get blind. So what we said, we wanted to see if we can use this model and use our cells to inject them into this hypoxic condition and try and prevent this pathological neovascularization. So in a way, stabilize this vascular. So the way we do that, we take the pups, and for 7 days, they just in atmospheric condition. Then we move them to oxygen chamber with high oxygen level for 5 days. And then on day 12, this is when we move them back to the atmospheric. So there is kind of mimicking of hypoxic condition by the movement from 75% to 21%. This is when we inject our cells. So these are retina images, and you can see here that in the in green is the CD31 positive cells or human CD31, and in red are the mouse lectin. And what we found, and here are more the quantification, that we can see that the IPS derivatives are localized into the area, the hypoxic retina, more than the human retina endothelial cells. Nicely, we could also find that the IPS derivatives really integrate with the mouse vasculature, as seen here with the yellow stain of the merge of the human of the mouse lectin, versus that the human retina endothelial cells really are not integrating. Rather, we see that the human vasculature is growing on top of the mouse vasculature. So Bria wanted to understand why this is happening, and she developed this nice assay when she took those collagen gels and she encapsulated in them SDF1 alpha, which recruit endothelial cells, and cultured the cells on top of those hydrogels. Then she put them in atmospheric and hypoxic conditions. And as you can see, the IPS derivatives, either atmospheric or hypoxic, really started to penetrate into the hydrogel responding to this SDF1 alpha. But the mature retina endothelial cells did not do so. So we looked into the specific receptor for the SDF1. This is the CXCR4. And we could see that in hypoxia is almost all our IPS derived endothelial cells express it, versus only 50% of the mature retina endothelial cells express it. Then we went back in vivo and we inhibit this receptor with antibody. And indeed, when we inhibit, you can appreciate it that we can barely see any green here, any green cells. Only mouse vasculature, versus this is the control when we injected cells into both. So with that, Bria came up with this nice idea that in a hypoxic condition, first of all, our IPS cells are able to proliferate. They are also able to be recruited into the vasculature and integrate with that. But throughout our studies, we realized that there is really a need in the next studies, is to focus on tissue-specific endothelial cells. So I told you the story about retina endothelial cells, and retina endothelial cells have specific barrier functions that are different from, of course, from endothelial cells in the heart and in the liver that have different jobs. So really, we think with iPS cells, we need to move forward and look into tissue-specific endothelial cells, and that really will allow us to understand more organ morphogenesis and organoids in general. Organ on a chip studies will be enabled by it, and towards a personalized medicine. So with that in mind, I showed you everything that we have done mostly in 2 dimensions, but when Kyung-Min Park joined the lab, I asked him if we can try and control oxygen level in 3 dimensions. So he came up with this nice idea of what we call oxygen consuming hydrogels, where he simply modified the backbone. In this case, it's gelatin. It can also be a polysaccharide with ferrolic acid. And by using laccase as the enzyme to polymerize, it's also consuming oxygen. So what's happening basically is that we have a hydrogel that is formed on the bench while consuming oxygen. If we modify correctly the thickness of the hydrogel and the level of media that we add and the concentrations of the cells, we can control the level of oxygen. Because we can control all of these parameters, we can compute and predict what the level of oxygen will be when the hydrogel is thinner, is thicker with media, sorry, thinner with just interfacing the air, the media with different level, different volume. And indeed, when we encapsulated endothelial progenitor cells, we could read the oxygen again in a non-invasive measurement. And you can see here, this is the hypoxic hydrogels, which are thicker and the same cell concentration in both hypoxic and non-hypoxic hydrogels. We can maintain hypoxia, which is here in this case is below 3% for over 48 hours. And this is the non-hypoxic hydrogel. We see some dip of the oxygen level, but then it's stabilized with the air penetrating into the hydrogel. So this was fantastic because to activate hypoxia-inducible factors, it should take about 4 to 6 hours for the transcription factor to accumulate with the nucleus and activate signaling pathways. So indeed, what we found is when we encapsulate these cells in the non-hypoxic versus hypoxic, we could see these nice networks of a vasculature form in the hypoxic hydrogels with no addition of any other growth factors. In this study, we could also find that what's happening is the hypoxia is stabilizing both HIF1 and HIF2, which upregulate matrix metalloproteinases that allow matrix degradation and then eventual vascular bed formation. And this was a great finding in 2 ways. That first, that's the first time that actually was a connection between endothelial cells degrading the matrices and the role of hypoxia in that. And the second, this was really a platform for us to study a more mechanism of the vasculature. And one thing we noticed in the literature, there was some evidence that circulating endothelial progenitors basically form clusters in hypoxia. So what happens, and this was reported in Lib ischemia and also in tumor, is that the endothelial progenitors are in the bloodstream, then they are recruited into the area of ischemia. So this is a hypoxic condition, and there they're forming these clusters, and then the cluster form networks. And you can see that also, it was also evident in tumors where people basically found these clusters of endothelial cells that later form network, and this network integrate with the tumor vasculature. So Michael in the lab asked, can we use this hydrogel system to study the mechanism? Because there was just reporting of this phenomena, but not really mechanistic understanding. So he took this hydrogel that I just told you about, but this time he doubled the cell concentration in both hypoxic and non-hypoxic. And in the hypoxic, he started to see this cluster form within about 8 to 10 hours, and this cluster basically stabilize and grow and eventually start kind of to sprout. And we could not see that in the non-hypoxic condition. And these are the non-invasive measurements for dissolved oxygen. So this is how it looks like in the culture. So over time, you could see that the cells start to kind of aggregate, forming these clusters. And eventually, these clusters will fall into the bottom of the hydrogel. You can see it coming right now. We could not see any cluster formation at all in those non-hypoxic hydrogels. So we asked, why is it happening? First of all, we looked into hypoxia-inducible factor 1. So we try to induce this hypoxia-inducible factor, but we still see no cluster formation. We try to inhibit this HIF1, and we still see this clustering forming. But when we inhibit the reactive oxygen species with a DPI, we see inhibition of cluster formation. We then said, well, is that really connect to protease or protease expression in those cells? And indeed, when we take these non-hypoxic hydrogel and we just add a specific concentration of MMP to degrade the matrix, we could see that that allows the cells to aggregate. So these clusters will stabilize over the 24 hours, and I hope you can see that with a Vic adhering between the cells and phalloidin. It's all organizing. Also the ICAM around the cells and integrin beta 2. So the clusters are real and are stabilizing over time. But then we really couldn't see any vascular network from that. And the reason was because the hydrogel became so soft because of those proteases. So Michael decided that we can, after 24 hours, we can secondary cross-link that with a transglutaminase to make this hydrogel a little bit stiffer. And when he did that, we could start to see this huge formation of huge networks that we typically don't see. This is what we would see. That's the sprouts that we would see in the control in the hypoxia. And these are basically these huge networks that we would see with those once we made those hydrogels stiffer with a stiffer hydrogel. We also confirmed this in vivo, where we injected those hydrogels in subcutaneously and examined if we have recruitment of endothelial progenitor cells in clustering. So the way we did that, we injected those hydrogel, we added the SDF1 and injected also fluorescent or GFP labeled endothelial progenitors. And you can really see that in the hypoxic condition, we have those clusters that we could not really see in the non-hypoxic condition. Still, understanding the pathway was very complex because what we have in those hydrogel is this nice, gradient of oxygen, which goes from low to high. But if we want to look into gene expression, it's very complex because here we have a gene expression in very low oxygen and here we have in higher oxygen. So when we did some experiments, we just couldn't find any differences. So Michael came up with this idea to generate these layered hydrogels where we can put the cells in what we call non-hypoxic or middle layer would be the moderate hypoxia and severe hypoxia in the lower levels. And with invasive measurement of dissolved oxygen, and this is with a microneedle, we can really find these 3 distinctive layers of oxygen or hypoxia regulation. So we went ahead and did a RNA-seq, and here is an example of the principal component analysis, which basically confirm the clustering of all our experimental conditions in the different time points. So you can see here a non-hypoxic and hypoxic after 40 minutes, after 10 hours, and after 24 hours. So I'm not going to go over all the gene analysis that we have done, but at the end of those analyses and some inhibition studies, we came up with this idea that what happens is that the EPC leave the bloodstream, they are entering this hypoxic condition, and this activates this reactive oxygen species and oxidative stress and apoptosis. What happens, the cells really need to cluster together to start to upregulate this cell cycle and cell survival and downregulate this apoptosis. Those clusters then have to stabilize, leading to more proteases and cell cycle and cell survival upregulation. And eventually in the dynamic matrix, it will stiffen, and in dynamic matrix stiffening, it would actually will start to sprout and make those networks. So with this, I'd like to go over one more aspect of studies that we are looking into, and this is vasculoelasticity. Specifically, a lot of studies have been done on this stiffness of the matrix, but less, or in more recent year, about the vasculoelasticity. So in general, viscoelastic materials will deform in response to the application of a load, and then they'll undergo this, what we call stress relaxation. So if you can see here with this nice schematic that one of my students, Eugenia, prepared, is when we have constant strain on the polymer. On the polymer, it starts to deform when we stop to apply the strain. Basically, there is what we call the stress relaxation, or the ability of the polymer or the matrix to go back to before the deformation. And in 2000, Sharon and colleagues published this nice review in Nature that showed, that kind of made the case for vasculoelasticity as an important parameter to study. And they show that this nice figure show that many of the tissues in our body are really vascoelastic. So Zhao Wei and Rahel Schellenman in the lab went to ask how vasculoelasticity impacting this tissue formation, or in our case, the vasculature. So what we have used is hydrogels that are both used from gelatin and dextran. And we use this dynamic covalent bonds and what we call conventional non-dynamic bonds to generate two types of hydrogel, what we call dynamic hydrogels, or more viscoelastic, and non-dynamic hydrogels. So first we confirmed that these have a specific stress relaxation and stiffness. So we could have this dynamic hydrogel and non-dynamic hydrogel either soft or stiff, and there is no difference in that. And you can see this over time in culture. This stiffness is still the same, but this hydrogel will have a different stress relaxation in each of the soft or the stiff matrices. And this difference in stress relaxation will remain throughout 3 days of culture. That was very important because that's about the time that it takes for the endothelial cells to make networks in culture. So we went to ask how endothelial cells mechanosense matrix and when they undergo vascular morphogenesis. So this is basically summary of how vascular morphogenesis is happening. We have the endothelial colony forming cells in our case. They will form, once we encapsulate them in the hydrogel, they'll form vacuoles and lumen, then they'll sprout and branch, and then make the network. And when we encapsulated these cells in the dynamic hydrogel and the non-dynamic hydrogel, we could see these beautiful networks in the dynamic hydrogel. And we could see some sprouting and maybe networks starting to form, but never really reaching a full a network formation with this time point of 3 days. And we could see those in the dynamic hydrogel. These networks have this beautiful lumen forming. So we went to see what is the molecular mechanism. We looked first into the vacuole and lumen formation, which happens around 4 to 6 hours after encapsulation, and we couldn't see really any difference between the two hydrogels. So then we hypothesized that what's happening is actually at this stage of the sprouting and branching, and that the cells that are encapsulated in the dynamic hydrogels are able basically to push away these hydrogel networks and sprout, which cannot happen or they cannot do in the non-dynamic hydrogel. So we looked into that, and first we did this reflection or traction force microscopy with the beads, both in dynamic and non-dynamic hydrogels, and we could see that the displacement of the beads as well as the speed by which they're moving is faster and further in the dynamic hydrogel compared to the non-dynamic hydrogel. We could also see that these cells are contracting while they're doing that in the dynamic hydrogel. We see that with the stain for phosphorylated myosin light chain. And you can see here, this is the stain and it's localized with the epheloidin, with the F-actin. And this doesn't happen in the non-dynamic hydrogel. We then continue and looked into cell contractility, how it's impacting focal adhesion. And you can see here with the dynamic hydrogel, we start to see, this is high magnification of this image, this clustering of integrin that then growing into this focal adhesion. So this is integrin beta 1 and the focal adhesion is stained with vinoculin. And this is both of these are also quantified here. And we also did a QRT-PCR, and we can see upregulation of both integrin beta 1 and integrin alpha V. So there is integrin clustering leading those to focal adhesion. And it's all through contractility, because when we inhibit cell contractility, we see basically no integrin clustering. And we see, compared to the control, we see downregulation of vascular networks. We continued with this pathway and we found that this leads to phosphorylation of the focal adhesion kinases, so activation of the focal adhesion kinases, again in the dynamic hydrogel, but not in the non-dynamic hydrogels. And we could see that when we inhibit the focal adhesion kinases, we could see again downregulation of or almost no vascular network formation. We see that the endothelial cells produce their own MMP, so they are degrading the matrix and this is with MT1 MMP stain and PCR for the soluble MMPs as well. Again, when we inhibit this degradation, this is a general MMP or I should say wide range MMP inhibitor, we could see no network formation. Finally, the cells start to produce their own extracellular matrix. With a qPCR, we could see here this collagen 4 and laminin, and you can see this nice collagen 4 in red around a luminal vascular network. So finally, we looked into that in vivo, and we inject the cells, the hydrogels with the cells, again subcutaneously. We could see the formation of nice network in the dynamic hydrogels. This is after a week, but we could not see that in the non-dynamic hydrogels. When we encapsulate the cells with no, encapsulated the hydrogels with no cells, we could see the recruitment or the angiogenesis of small vasculature into the hydrogel in the dynamic hydrogels. This is with the brown stain here. But we could see only small vessels, kind of the interface between the tissue and the hydrogels in the non-dynamic hydrogel. So very a little amount of vasculature penetrated into this hydrogel. We also know that the vasculature is functioning, because when we inject even blue into the tail vein, we could see this the dye penetrating into our dynamic hydrogel. So with that, we proposed this mechanism by which vasculoelasticity regulate vascular assembly. So the dynamic hydrogel networks allow basically the cells to contract and push the networks away. This contractility activates this integrin clustering that activating focal adhesion kinases, MMP expression, so degradation of matrix, but also new matrix formation, and eventually leading to a morphogenesis. So with that, I'd like just to summary that I showed you that oxygen tension regulates vascular fate, network assembly, and integration, that there is a major role or interactions between hypoxia and extracellular matrix, and those interactions guide neovascularization. And what I've showed you was mainly in a ischemic condition, when we try to understand that also in a cancer. And lastly, I showed you that hydrogel network dynamic regulate vascular assembly. So with that, I'd like to thank, and I thank throughout the presentation to the students that performed this study, but these are the current students in the lab that just moved to Duke, with all our previous and some current collaborators, the funding agencies, and of course I have postdoc positions open because we just moved, and I'll be happy to take any questions.
Thank you for the great talk, Dr. Gerecht. Appreciate it. Very nice work about the fundamentals. Couple questions. You tried hypoxia. Have you tried to increase the oxygen concentration the other way? For instance, cancer cells to grow need hypoxia. Can you increase the oxygen to see if it can reverse it?
We actually didn't try that in the we didn't try that in the context of cancer. We tried that in some of those studies. So our control is actually when we encapsulate those hydrogels with oxygen forming particles. So this is our control also, both in vitro and in vivo. So we tried it, but we never looked into above 21%, if that's what you're asking.
Yes.
Yeah, we never looked into above 21%.
The other one is the anastomosis or integration. How long does it take? You've seen nice, the structures that integrate with the vasculature of the tissue.
Yeah, so I think the first events of integration, we notice after 7 days. If we want to see more robust, we should wait for two weeks. But I think integration is much faster. Otherwise, it wouldn't have survived. We're just unable now with our imaging to be able to find it.
I see. You also mentioned about colonies. Would the same concept apply for single cells or maybe 10 cells, or do you have any thoughts about that?
You mean in terms of what?
Reacting to oxygen environment. You mentioned a couple slides about colonies of cells.
Right.
And they're behaving, but I'm just wondering if it's 1 cell, 5 cells, what does a colony mean, and does it have the same response?
Right. Yeah, that's a good question, actually, because what we found is that with these colonies, the cells kind of survive, right? They come together and they survive in these hypoxic conditions, and then they are able to make those networks. I don't know, that might be the case in other cell types. We just know that, endothelial cells eventually will form these networks. If cancer cells do that, they survive and they just grow, but it's definitely possible.
I see. You also mentioned about these dynamic gels.
Yeah.
Can you a little bit elaborate, because there's other types. People are working on self-healing gels, people using organs on a chip, mechanical stress on the gels. What is your current research on dynamic gels, and what is your future plans?
Yeah, so currently we're actually moving into cancer. So the original work, by Mooney and Chaduri in Mooney in Harvard and Chaduri in Stanford is about cancer cells migration and some stem cells. So we're moving now to look into how cancer cells respond to these hydrogels, and are they growing, are they proliferating, are they migrating more, and also how they're interacting with the tumor vasculature. So these are kind of the directions we're going with that specifically.
Thank you. Thank you. Can the viscoelasticity of hydrogels be modified in a way to prevent angiogenesis and epithelial cancer?
Yeah, I hope so. I think this is a good question. We work on, we try to do like co-encapsulations of cancer cells and endothelial cells in those hydrogel and see if we could, understand how it's impacting extravasation or intravasation. But I don't know yet. It's a good question.
Thank you. Thanks for a wonderful talk. Does stress relaxing hydrogels also increase assembly of cells endogenously?
So I'm not sure I understand what does that mean, cells endogenously.
The cells, would the cells assemble in these relaxing hydrogels?
In this relaxing, I think it's increasing, but we actually didn't look into that specifically. So I can't comment on that. I don't know.
Okay, thanks for the great talk. Have you ever checked the vascular genesis of early vascular cells in comparison to HUVEX? Due to the formation of large cluster of cells, do you see more vascular network at the bottom of the gels?
Yeah, so for the first part, we try to work with HUVEX, but they're very finicky for us to make robust networks. So I would say that the answer would be yes, the endothelial colony forming cells always perform better. And the second question, eventually the cells will, if we're not going to stiffen the hydrogel, so they're making the clusters and then they're producing all these proteases, and if we're not going to stiff the hydrogels, they will fall on the bottom of the hydrogel, and we don't analyze them. So for them to make networks, we are allowing the cluster to form, we stiffen the gel, and then they're forming the network. So anything that falls to the bottom, we basically don't, it's out of the focus and we don't analyze that.
Okay, thank you. In the traction force measurement in the dynamic gel, have you considered the time dependence in the calculation?
Could you repeat that, please?
Sure. In the traction force measurement within the dynamic gel, have you considered the time dependence in the calculation?
Yes, we considered that, and we also did it in a specific time or window in the encapsulation of the cells. So it's right when we encapsulate the cells, we looked into the bead movement and not later on, because once the cells start to form the network, there are other forces in play that we're not considering.
Okay. Can we apply a magnetic force to treat blood flow microenvironmentally in capillaries by spions?
I have no clue. We never work with magnetic forces.
I see. Thank you. Let me see. A couple other questions came in. Is it possible to use trophoblast cells as the cells which do the first angiogenesis in embryo that develop the chorion later to treat myocardial infarction?
We never worked with those cells, but I know there are groups that actively work. I don't know if they tried in the cardiac. I actually haven't heard about that, so I don't know.
Let's see. A final question: how is the hypoxia working from a regenerative medicine perspective?
Yeah, so regenerative medicine perspective, you think about, I don't know, I'll take the most simple example is the wound healing, right? So if you have injury and the wound, the vasculature is not functioning. So it is hypoxic condition. Of course, if the wound healing is on the surface, then there may be a oxygen penetration. But basically, every organ that doesn't have any vascular in it is in hypoxic. So our thought is that we study how those hypoxic conditions will impact the regeneration of those or generation by these cells.
I see. Well, thank you so much. That's all the questions I have. Thank you for your time, Professor.
Great. Thank you so much for inviting me.
Thank you. Have a nice day.
You too.